Effects of insufficient sleep on circadian rhythmicity and expression amplitude of the human blood transcriptome by Mushtaq Abdi and Amal Ali. Insufficient sleep is when an individual fails to achieve enough sleep to support adequate alertness, and there are two types. Acute sleep loss refers to temporary reduction of sleep, which can be no sleep or reduction of the required hours for a day or two. On the other hand, chronic sleep loss is when an individual regularly sleeps less than the normal hours required for optimal functioning. Chronic sleep loss can cause many health problems such as cardiovascular disease, obesity, cognitive impairment, and diabetes. Circadian rhythmicity. A circadian rhythm is usually referred to as the body's internal clock. It's a mental, physical, and behavioral change that follows a 24-hour cycle. They are generated endogenously, however they respond to light, darkness, and temperature in an organism's environment. They are found mostly in living things such as plants, tiny microbes, and animals. They are most important in animals as they determine their feeding routine. The study of circadian rhythms is called chronobiology. Transcriptome is the sum of total of all messenger RNA molecules expressed from the genes of an organism. In other words, all the transcripts in a given cell. Therefore, by analyzing the transcriptome, we find out when and where the gene is turned on and off, and gene expression, which is the amount of gene activity, and in this case, the effects of insufficient sleep on gene activity. The aim of this study was to identify the effects of insufficient sleep on the circadian rhythmicity and on the human blood transcriptome. The experiment consisted of 20 healthy participants that were exposed to 1 week of insufficient sleep of 5.7 hours in total, which resulted only in 0.03 in 24 hours. Also, they were exposed to 1 week of sufficient sleep of 8.5 hours. After each condition, 10 whole RNA blood samples were collected from each participant, and the effects of light activity and food were also controlled during the period of total sleep. In addition, to measure the effects of insufficient sleep on the whole blood transcriptome, RNA sampling was conducted. Therefore, to assess the effects of insufficient sleep on the whole blood transcriptome, frequent RNA sampling was conducted over more than one complete circadian cycle, using an established constant routine protocol to control for these confounding factors. Mixed model ANOVA for repeated measures revealed the main effect of sleep condition, sleep restriction phase control, on the levels of transcripts encoded by 711 genes. 3.1% of the genes determined as present in the arrays. Of these genes, 444 were downregulated and 167 were upregulated following sleep restrictions. The two genes that were most significantly affected by sleep conditions were MFNG and DCAF5, which were downregulated in response to insufficient sleep but had not previously been directly implicated in sleep regulation or circadian rhythms. Genes related to circadian rhythms and sleep, which were downregulated after sleep restriction, included RORA, PER2, and 3. Prevalent circadian genes were defined as those targeted by probes that showed a significant circadian oscillation in transcript levels in the number of participants that resulted in false discovery rate, FDR, of less than 5% in each condition. Assessment of each individual expression profiles for a prevailing oscillatory component with an approximate 24-hour period in the control condition identified 8.8% circadian genes, which included PER1, 2, and 3, and other genes that associate circadian rhythms, sleep and metabolism, and other variables. In this balanced crossover design, as seen in Figure 1, participants obtained an average of 5.7 hours of polysomnographically assessed sleep per 24 hours during the 7 nights of sleep restriction condition, and 8.5 during the 7 nights of control condition. Sleep obtained in the sleep restriction condition was not sufficient to maintain alertness and performance. On the last day of sleep restriction, participants were significantly more sleepy, as seen in the Karolinska Sleepiness Scale, 4.3 versus 3, and had more lapses of attention. To identify the transcripts that responded to total sleep deprivation, the cumulative trend for each series was calculated and identified probes with statistically significant upward or downward trends during the constant routine after both the control and sleep restriction conditions. The prevalent time and weight-dependent genes are defined as those targeted by probes whose transcript levels showed a significant trend in response to total acute sleep deprivation in a number of participants that resulted in an FDR of less than 5%.
The analysis identified 122 time-awake dependent genes during the total sleep deprivation following the control condition. Following the sleep restriction condition, there was a seven-fold increase in the number of prevalent time-awake dependent genes, 856 genes compared to the control condition, which had 122 genes.
Circular organizing map (SOM) was used to identify distinctive temporal patterns with a set of prevalent circadian profiles in the control condition. This analysis identified median circadian expressions at profiles that separated into 5 clusters with peak times ranging from late in the biological day, cluster 1, the biological night, cluster 2 and 3, and the early and middle of the biological day, cluster 4 and 5.
Well-known circadian sleep genes in cluster 1 included RORA, NFKB2, CSNK1E. Genes whose transcript peaked during the biological night, clusters 2 and 3, included sleep and circadian regulated genes such as PER1, 2 and 3, and many other genes.
This study has demonstrated that insufficient sleep to an extent frequently or chronically experienced by many individuals in industrialized societies alter the temporal organization of the human blood transcriptome, including the circadian regulation of the response to acute sleep loss.
The biological processes encoded by the genes most affected by sleep restriction included chromatin organization and modification, gene expression, inflammatory and stress responses, as well as cellular macromolecule metabolism and oxidative stress responses.
The sleep restriction protocol led to a reduction of post-sonographically assessed total sleep time, 5.7 hours per 24 hours, which is insufficient for this age group, as evident from the increased sleepiness and lapse of attention compared with the well-rested control condition.
Insufficient sleep of this magnitude and duration may also lead to changes in many metabolic and endocrine variables. The current protocol was designed to assess effects of sleep restriction on the temporal organization of the blood transcriptome, independent of the direct masking effects of longer light exposure and more intense sleep, which are present during the imposition of restricted sleep.
To quantify the effects of insufficient sleep on the endogenous temporal organization of a transcriptome, we assessed this during a period of sustained wakefulness and aligned the transcriptome time series with the rhythm of plasma melatonin. The melatonin rhythm provides an internal circadian phase reference point that is generally relevant because of the circadian variation in considerable proportion of the transcriptome, and particularly relevant because sleep restriction has been reported to shift circadian rhythms as well also observed in this study.
Because the constant routine conditions involve a period of sustained wakefulness, the protocol also allowed the assessment of the effects of acute total sleep loss on the transcriptome, which has been the focus of many rodent studies. Importantly, the analysis of the transcriptome revealed that the effects of sleep history, circadian phase, and acute total sleep loss interact.